Okay, I'll begin by thanking the organisers for this opportunity. So it's a very broad title. I tried to keep it broad because it was so much in advance. So today in my talk today, I'm going to talk about the genetic, the epigenetic and external environmental factors influencing aging and its association with health. And we're also one thing missing from this title is the epigenetic clocks. So I'm going to explore the utility of epigenetic clocks to assess biological aging. So let's begin. So aging is a very complicated process involving multiple factors. But why are we interested in aging? Because we're all aging. And one of the reasons we need to, or we want to understand aging is because it's associated with several diseases. And here are just a few listed. So, there's an increasing body of evidence that associates epigenetic and genetic markers such as cellular senescence, telomere shortening, and genomic instability with aging. But not only are they associated with aging, but they also collaborate to drive the process of aging. So for those of you who are unfamiliar with the term epigenetics, well, I'm not going to explain what it is here because it's too big a topic to cover. But essentially, it's the molecular switch which responds to environmental cues. So your body is listening to what's going on outside and it responds internally biologically. So this is done through sorry, don't know what's happened. Attaching a methylation group. So I'm interested in DNA methylation. There are other kinds of epigenetics as well. The important thing is that epigenetics involves environmental factors. These include lifestyle, diet, where you live, your life's experiences, pollution. The difference is that the epigenome links the genetics information to the phenotype without altering the genotype. So it changes the transcriptomic profile without altering the DNA sequence. Another important factor is that by doing this, so unlike genetics, this process can be reversed. So if you identify a risk factor, say living in a polluted areas, you're more at a higher risk of developing an autoimmune disease, then you could develop policy to limit the pollution, and that can then reduce your risk. Okay, I keep pressing the wrong button. So we mentioned it links the genotype to the phenotype, and until recently, we had no way to assess the biological age. That's until 2013, a paper was published, which introduced the concept of epigenetic clocks. And you have to be careful. So this was, again, a machine learning method, which was trained on DNA methylation data, and they identified a set of sites that correlated with age, so they were able to predict accurately an individual's age. So, and this is widely used now in aging and cancer research. And then since then, so this became known as the first generation of clocks. So this was a Horvath clock and the Hannum clock, which was blood specific. So we know that our bodies are aging at different rates, and different organs are aging at different rates. So there are different clocks for different organs. And the second generation was trained on, so it's called PhenoAge, was introduced morbidity. So it was trained on blood markers like CRP and creatine and lymphocyte counts, etc. Getting back. And this was further built on by Grimage, which introduced life experience, lifestyle factors like smoking, how if you smoke or not. So these clocks were then trained on someone's experiences as well. And there's a third generation which measures the rate of aging. And now there are clocks being developed for single cell. So we mentioned the epigenetics can be reversible. So the clock score, that refers to, so if you're on an accelerator, if you're living in a polluted area, you smoke, you don't do any exercise, the chances are that your internal biological clock is probably higher than your chronological calendar age. And however, if you do then start exercising, you introduce healthy diet, then that can reverse the clock, and then your internal biological age will be younger than your actual age. So David very nicely set the scene for me for the importance of sepsis, so I don't need to explain why it's important, but I use this as an example. So it's in critical care, it's very difficult to discriminate between those patients who are going to develop sepsis versus those who are not. And we wanted to, and aging is a risk factor, so an older person is more likely to have poorer outcome of either critical illness or sepsis. So, you know, sepsis is underpinned, it's inflammatory response in response to an infection or an injury. And so the body then starts attacking its own organs. So there's a shift in the pattern is due to changes in epigenetics. So we wanted to understand if those changes in epigenetics is also linked to the process of aging. So can we determine the epigenetic age of these people? So we looked at a study. This is a Canadian study. These patients were admitted into intensive care units, and the bloods were taken on day one. So they were matched for severity, comorbidity, age, and sex. And then we also had outcome data as well. So I've got a lot of slides to go through, so I won't talk you through all the results, but basically what we found was that the septic patients had an increased epigenetic age versus the critically ill patients. And this was significant using both the first generation and the second generation clocks. And then we noted a difference between survivors and non-survivors. So that the non-survivors, those that died, had an increased epigenetic age in both the septic patients and the critically ill. So then this work's been published, and then so this raises the possibility of using this to discriminate between the critically ill patients that may develop sepsis. And then more work needs to be done to verify if the utility of epigenetic clocks and to determine if it outperforms the current clinical scores that's being used in the clinic. What I do want to do is acknowledge lots of people who've helped with this work. So thanks, and I'll take questions.